First, talk about female gametogenesis, which is also known as oogenesis. Primordial germ cells from the wall of yolk sac arrive in the ovary at week 6 of embryonic development and differentiate into oogonia. Oogonia enter meiosis I and undergo DNA replication to form primary oocytes. All primary oocytes are formed by the 5th month of fetal life and remain dormant in prophase of meiosis I until puberty. During a woman's ovarian cycle, a primary oocyte completes meiosis I to form a secondary oocyte and a first polar body which probably degenerates. The secondary oocyte enters meiosis II and ovulation occurs when the chromosome align at metaphase. The secondary oocyte remains arrested in metaphase of meiosis II until fertilization occurs. At fertilization, the secondary oocyte completes meiosis II to form a mature oocyte and a second polar body. Hormonal control of the female reproductive cycle. The hypothalamus secretes gonadotropin releasing factor. In response to GnRH, the genohypophysis secretes the gonadotropins such as follicle-stimulating hormone and luteinizing hormone. Follicle-stimulating hormone stimulates the development of a secondary follicle to a graphene follicle within the ovary. Granulosa cells of the secondary and graphene follicles secrete estrogen. Estrogen stimulates the endometrium of the uterus to enter the proliferative phase. And luteinizing hormone stimulates ovulation. And following ovulation, granulosa lutein cells of the corpus luteum secrete progesterone. And then progesterone stimulates the endometrium of the uterus to enter the secretory phase. Male gametogenesis, which is also known as spermatogenesis, is classically divided into spermatocytogenesis, meiosis, and spermiogenesis. In spermatocytogenesis, primordial germ cells from the wall of yolk sac arrive in the testis at week 6 of embryonic development and remain dormant until puberty. At puberty, primordial germ cells differentiate into type A spermatogonia. Type A spermatogonia undergo mitosis to provide a continuous supply of stem cells throughout the reproductive life of the male, called spermatocytogenesis. Some type A spermatogonia differentiate into type P spermatogonia. In meiosis, type P spermatogonia enter meiosis I and undergo DNA replication to form primary spermatocytes. Primary spermatocytes complete meiosis I to form two secondary spermatocytes. Secondary spermatocytes complete meiosis II to form four spermatids. In spermiogenesis, spermatids undergo a post-meiotic series of morphological changes called spermiogenesis to form sperm. Newly ejaculated sperm are incapable of fertilization until they undergo capacitation, which occurs in the female reproductive tract and involves the unmasking of sperm glycosyl transferases and removal of proteins coating the surface of sperm. Male infertility. It depends upon sperm number and motility. Infertile males produce less than 10 million sperm per ml of semen. Fertile males produce from 20 to more than 100 million sperm per ml of semen. Normally, up to 10% of sperm in an ejaculate may be grossly deformed, but these sperm probably do not fertilize an oocyte owing to their lack of motility.